We're good.
So this is biology. My name is Connor Murphy. My ID number is 1520517, and we're going to talk about mouse cancer. In this question, it talks about basically trying to treat cancer in a mouse, and something goes wrong. So our first method we use is radiation therapy. So this is a pretty good method to treat cancer cells because it basically forces apoptosis in a cell. How does it do that? Radiation therapy, essentially, the concept behind it is that it does damage to DNA in a cell, and it basically breaks up the DNA. And so when DNA breaks up, it acts as an apoptotic signal. So very conveniently, I have down here what happens when an apoptotic signal is released. So when DNA damage happens, it acts as an apoptotic signal, like I said. And when that signal is released, something special happens in the mitochondria. So here's the mitochondria right here. This is the intercellular space, or intermembrane space here. These are membrane proteins. These are called BH123 proteins. And these are called cytochrome C proteins. So these are going to be very important for cell death. So when that apoptotic signal is received, when the DNA damage happens, essentially, what happens is these BH123 proteins form pores. So they basically bind together and form a pore. And what this pore allows for is the release of these cytochrome C molecules to leave the matrix. When it leaves the matrix, it binds to APAF1 proteins, and the structure looks something like this. This here is a card domain. So this structure here finds a bunch of other structures which happen because cytochrome C just evacuated the mitochondria, and they form this flower-like structure here. This is called an apoptosome. So all the card domains basically form in the centerpiece, and they act as adapter proteins. So when the structure forms, it brings these rectangles I've drawn beforehand. These are called caspases. This is caspase 8 specifically. So what happens is these caspases bind to this apoptosome, and it basically self-cleaves when this happens, and it forms a tetramer, which is this. And this caspase, this is a caspase, cuts executioner caspases. And these, basically they self-cleave, so normally a caspase looks something like this. Ignore this for now. Just focus on that. So these self-cleave, and then it becomes this structure here. So these bottom parts go like that, and then it can cleave executioner caspases. So executioner caspases basically begin the process of cell death. So that's why we use radiation therapy to begin. So what happens next? So we notice that this therapy is no longer effective, and the tumor continues to grow rapidly. Essentially, we find out that there's a new loss of function mutation. So a loss of function mutation. So a loss of function mutation is a mutation. Basically, it changes a protein so that it stops working. So the protein that stops working is related to P10 gene. So right here, this is the protein associated with the P10 gene. And so it acts as a phosphatase for a membrane protein called PIP3, which turns it into PIP2. So why would that affect our mouse? Okay, so first let's talk about FGF. FGF is a signal protein. So I've drawn them up here beforehand. And so the signal protein will come down and bind to this receptor in the cell. And this receptor is called RTK. So receptor tyrosine kinase. And what's cool about this kinase is that it autophosphorylates. So when this FGF binds, it brings the tyrosine kinases, which are kind of throughout the cell, it brings them together, and it autophosphorylates and activates itself, essentially, as a dimer, which is basically this dimerizes this receptor. So what does this receptor do? It basically activates this thing called PI3 kinase. And this PI3 kinase interacts with this protein here and this protein. So think of PI3 kinase as the antagonist to this mutated form. So what does this mutated form do? This mutated form takes PIP3 and turns it into PIP2. So that's what that does. We can ignore that for now. This PI3 kinase turns PIP2, turns it into PIP3. So what does PIP3 do? Why is it so special, and what does it have to do with our mouse having cancer? So PIP3 acts as a docking site, so for another protein called PDK1 and AKT. Okay, and these are membrane proteins, and basically acts as docking sites. So when it brings them up, this, when it binds, phosphorylates this. So let's draw a P here to signal that it's been phosphorylated. And this protein, which is kind of floating in the cytosol, phosphorylates this again. When it's double phosphorylated, this detaches from the membrane. It kind of floats around and binds to this, which is a BH3 protein. Specifically, it's called BAD. BAD is normally bound to BCL2. So what does this have to do? So when it binds to BAD, it releases BCL2 into the membrane, which is why I've drawn it here. So what does this have to do with cancer? Well, BCL2 has a very interesting anti-apoptotic purpose. So what BCL2 does is that remember these pores here? It basically binds to these proteins and prevents them from making this pore. So let's kind of bring it all together. So initially, radiation therapy was working. So basically, it would go through, the DNA damage would happen, it would act as an apoptotic signal, it would trigger the mitochondria to open these pores, and these cytochromes would activate into a various signal. Basically, it triggers a cascade which causes the cell to die. Something changed in this molecule, which turns PIP3 into PIP2. So basically what happened was this is constantly keeping this pathway on. What does this pathway do? This pathway stops apoptosis. It binds to these pores and it stops cytochrome C from leaving the cell, which essentially makes our radiation therapy ineffective. Thank you.